course, there's DNA. So, as I mentioned, we're diploid. That means we have 2 chromosomes, homologous chromosomes. 23 pairs, 46 total. Each genome, all the genes are contained in every cell. Now, of course, these complementary chromosomes, we get one from mom, one from dad. So this is a homologous chromosome. The only difference between one side and the other might be different alleles of the same gene, different variants of the same gene. And each chromosome is a long DNA molecule. And the genes are, I guess, the functional regions. The genes make proteins, and they also make a whole slew of RNAs to control how genes are regulated. There are 4 base pairs. That's all. 4. So the outside DNA is actually a sugar backbone. It's got these base pairs. Let me, now let's go on. Okay, so we do okay, and let me make this bigger. Okay. That's an A. That's a T. Adenine, thymine. And then there's a oh, come on. That's a guanine. And this is a cytosine. These are bases. These bases are the things that actually tell your cells which amino acids to actually make, which then make up proteins. G and C always go together. A and T always go together. You'll never find an A and a C together. It doesn't work biochemically. So these are the instructions, but I mentioned the environment. You really can't have proper gene expression without an interaction with the environment. So I'll give you an idea of what I mean. You would think that developing a heart would be almost all genes, right? I mean, you develop a heart, you have all these proteins that are working, and it's like a blueprint. But it's not. It turns out that the heart develops in a specific environment. They've done some work with zebrafish, and they've looked at the circulatory system, and I don't know how many cells, probably, thousands of cells in the embryo, and a circulatory system starts to develop without a heart. But there's some flow going through it. And this flow actually causes shear going by the arteries. And so on one side, there's more gene expression because there's more blood flow. On the other side, there's less. And it causes indentation. And eventually, this indentation, because of these different shear forces from the blood, actually form the heart. There's all these genes that go on in forming a heart. All sorts of different things. Growth factors, just a slew of stuff. So you can see there, that's kind of like a cellular environment or molecular environment. You won't get a proper heart developing without that kind of environment. It just won't happen. So even the most simple thing where you think, oh, this is all genes, nah. It's always a gene by environment interaction. When we talked about wolves with long hair and thin hair, that's also a gene by environment interaction. First of all, cold weather causes gene expression and denser fur. So, like wolves in the summertime have lighter fur and winter denser fur. But in terms of evolution, it's always a gene by environment interaction. They've sent all these things up to space doing interesting experiments. And there's a lot of things that don't develop properly in 0G. So we just take gravity for, this is what we, evolved to deal with a certain amount of gravity. Well, if you took, let's say, a human zygote and let it develop in a petri dish up in space, it's likely that it wouldn't develop properly into a human being because there's zero gravity. So here we are. These are the different nucleotides, as I mentioned. I guess there was no reason for me to do it, but it's better. So you have those backbones made of sugars and phosphates. And then the rungs are these bases that I talked about, AT, CG, and there's nitrogen. And this is what holds the genetic code. So I mentioned ATGC. This is what it looks like. If you were to put, let's say, an A and a C together, they wouldn't work biochemically. So you get these different rungs, and to make an amino acid, what you do is you do a codon. Okay? So this codon is TGC, and it makes a specific amino acid. There's 20 amino acids there's more than that, but there's only 20 that life on Earth actually uses. So, anybody know? I guess you can see who discovered the structure of DNA. This was 1953. They got the Nobel Prize for it. At this time, people didn't know exactly what molecules genes were made of. There were other ideas that perhaps you start with a protein, And then there were people working on DNA, but it was very difficult to figure out the structure. And these guys actually built a model. Because they knew biochemistry, they knew how things went together. At the time, there were also competing ideas about what DNA looked like, including a triple helix. There was a guy at Caltech named Linus Pauling, who had already won a Nobel Prize in chemistry, who was working on it too. And then there was this conference in London, and there was this woman, her name was Franklin, and she did X-ray crystallography. And actually, with X-ray crystallography, you can actually see in the kind of like individual atoms of what molecules look like. And when these guys saw that, they suddenly realized that it was a double helix. Pauling wasn't there because he was considered a communist, so they wouldn't let him travel. He probably would have beat him to it. But anyway, these guys are the ones that figured it out. And then Crick later went on to actually show how you read the DNA code in terms of A, T, and GC. And codons, that means you get these different rungs. So DNA, the reason it's such a great molecule is that, well, you can copy it. Now, RNA is only single-stranded. Probably on life on Earth in the beginning, it was probably an RNA world, but RNA accumulates too many mutations. DNA is a lot more stable in that regard. And so this double-stranded DNA let's see, kind of unwinds. And so you've got complementary strands. It's DNA enzyme polymerase. And then what it does is each of these two strands is made into a DNA molecule again. So that you can produce two new DNA molecules that are each identical to the other strand. They're complementary, except for perhaps a mutation. And then there's this whole system that goes up and down the chain to make sure that there were no errors. And if there are, it goes and fixes them. Okay, to this. Going to learn more. So we've used this DNA analysis with these short tandem repeats for all sorts of things. A plane goes down, crashes. You've got body parts all over the place, and you want to know who belongs to who. So you do a DNA analysis on them, and then you get blood from, let's say, relatives, and you can actually see who's a match. So at the World Trade Center, same sort of thing. There was all these people, there were sometimes only a finger left, whatever, and in the end they could pretty much identify everybody that they had. You can use it to solve crimes, of course, like rapes, things like that. You can missing persons, and you can look at relationships in terms of paternity, child abductions, or you get some blood from someone. They're not in the database, but they're a very close match to a brother who's already in the database. And then it looks like, oh, it looks like it's this guy's, perhaps his brother. And then, of course, hopefully you can get blood from the parents, but they might not agree to it or something like that. So we can use it for all sorts of identification routines. So we can find it in a lot of different places. We leave cells behind wherever we go. I'm probably leaving DNA on this straw, and perhaps they'll use this against me because I've committed a crime. I do a lot of crimes because I find I get a kind of a rush to do crimes. I don't know. Anybody else in here do crimes? Yeah? Oh, good. Okay. We should get together later on. Okay. They don't pay me much here, so I have to go rob banks. I don't do local banks. It's like hunting your home covey of quail. You just don't do it. It's not looked upon as a proper etiquette. I usually go to Nevada, because I used to live there. To Winnemucca or Ely. The thing is, the trouble with that, this is off the subject, but the trouble with Nevada is everybody owns a lot of big guns. So, I mean, it's just, I'm thinking of a different, safer place, like Delaware, maybe, or something, I don't know. Anyway, so we can get skin from under a victim's fingernails, saliva from an envelope or a straw, clothes, whatever. And hopefully we can get these short tandem repeats and determine who's who. And of course, they've been using this recently to show that a lot of people they put in jail were the wrong people. Happens a lot. I'm sure they've executed the wrong people, but you know, what's the big deal? They're going to die. They'll make it all right, right? So, people just love to execute other people for some reason. Okay. Now, the thing is, let's say you get, in the past, you needed a lot of DNA to get enough material to get a good read for these short tandem repeats. But then they came up with this what's called a polymerase chain reaction, where you're basically making copies of the section of DNA over and over and over again. So even with a single piece of DNA, you can make millions of copies. And then you can actually look and see what the base pair, how many repeats they have to actually determine who's who. So this is kind of how it works. They can amplify the DNA using just a small amount of DNA. Let's say you only have, I don't know, 100 fragments of DNA. You can make it into millions. So with a PCR, you start with 1, you make 2, you make 4, you make 8, 16, and then all of a sudden you're making millions. So here we are, these variable regions, these short tandem repeats that I was talking about. And they're variable in length. And they're different between people. Now, if you're only using one STR, one region, you wouldn't be able to tell, because there's going to be someone with, let's say, 18, someone with 14, another one with 14, someone with 13. But if you take, let's say, 10 of these different regions, the chances of one person having something that's 14 on one and 55 on another between people is really small and remote. So that's how they actually do it. So here we look and we see 1, 2, 3, 4, 5, 6, 7 different variable regions. Okay, and we looked at this already. So the primers, which are on either end of this repeat, they're these special universal primers that can attach to complementary strands. And these are the repeats that you then measure. And you can use the same primers for different people. So here we are. This thing again. Person 1, 2, 3, 4, 5, 6. Everybody has 2 alleles, one for mom, one for pop. So this is the electrophoresis. You make, you put, let's see, these, let's see, into wells. So you take a little pipette, put a certain amount of the DNA into a well. In this case, you could do 5. Then you apply an electrical current. And these things, as you can see, negatively charged DNA moves towards the positive electrode. So DNA is negatively charged. And then the fragments move at different speeds. Because some fragments are bigger and they move more slowly, and other fragments are smaller and move more quickly. So the ones that are smaller, of course, are right down here. Okay. We can look and see what it looks like. So here are these different sized fragments again. So which STR is smaller? Anybody? What do you think? What? Which of the, let's say on person 2, which fragment is a smaller STR? The one on the top or the one on the bottom? Yeah, exactly. Smaller STRs move more rapidly. Okay, so this one is substantially smaller than this one. Here we have 2 that are almost the same size, but not quite. These 2 up here are both fairly heavy, pretty long. Okay, so we're going to do a little fun thing here. We've got mom and baby. Which band does mom and baby share? One in the middle, right? Now, of course, everybody has 2 copies, and baby has, they share, here we go. So, that's mom, and that's the baby's identical STR from mom. So here's baby, here's mom. Okay, now, Baby, oh, who's not the father? Oh, okay, so baby has this, right? So this is possibly the father, or this is possibly the father, but who's not the father? Well, baby doesn't share any alleles with the person who's not the father. So we've at least narrowed it down to 2 suspects. And remember, though, that we're using a bunch of different STRs, usually 13 or 14, and so we'll be able to differentiate between number 2 and number 3 about who's the real father. So as I just mentioned, when you do a DNA analysis like this, the more STRs, the more variable regions that you actually test, you narrow down the chances of focusing on one individual versus another. Okay, so who's the father? Doing this again. Now, mom and baby share that one. Here we are again. And now we want to know, does baby is number 1, 2, or 3 the man behind the magic curtain? Exactly. So, Papa okay, good. So the human genome that we have contains thousands of STR markers. Usually we use 13, sometimes more. You can detect, obviously, gender because of sex chromosomes. Female is XX, male is XY. And then if you use a standard set of markers, you can share the information across databases. They might be using some things that are called SNPs, single nucleotide NSP. What is it? Single nucleotide. I don't know what it is. So they're a little bit better. And then, in terms of, you can get a DNA profile. You have 13 different markers. The probability of 2 unrelated people sharing the same DNA profile across all 13 markers is 1 in a million, probably less than that. Oh, okay. Here's a question now. Anybody in here an identical twin? No. Anybody know an identical twin? Okay. An identical twin can go out and commit a crime and not get convicted, unless there's some other evidence to point to an individual. If it's just this stuff, there's no way to tell between one individual who's an identical twin and another. So you can tell your friends if they want to commit crimes, go ahead. Because there has to be a reasonable doubt, and in that case there's some reasonable doubt. Okay, I think we'll go